Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. We invite you into the stories, struggles, and breakthroughs shaping the future of human health. From cutting-edge brain science to discoveries transforming how we heal, adapt, and thrive. We explore the ideas that matter and the people behind them. Together, we look across diseases, technology, and research fields to understand not just what is changing, but why it matters for patients, families, and the future we all share. Join us as we uncover new insights, spark collaboration, and illuminate the science that can improve our lives. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide to the future of health science. Joining me today is Micaelly Alves, a PhD candidate at Temple University. She is also a 2025 Toffler Scholar. We will be discussing Micaelly's focus on cerebral amyloid anthropology. So welcome, Micaelly.
Thank you.
This is great. I'm really excited to share your story. You're a Toffler Scholar, and we'll talk about that later. But a couple of weeks ago, when we were chatting with you about your grant and what you were going to do with it, it was just amazing that you have such a great background. And I wanted to kind of share this and highlight you and your growing career. I would like to start with, you've grown up in a bunch of countries, and I would love to just tell everybody about that, different cultures, different kind of languages. How did this shape how you see the world and really why you chose research?
Right. So I was born in Philadelphia, which is also where I live today, and I think it's kind of just a coming back to my roots in a way. So I was born here and I lived here until I was about 8 years old. Then I moved to Brazil. Both of my parents are Brazilian. My entire family is Brazilian, so Portuguese was very predominant for me growing up. And one of the reasons why my mom actually decided to move back to Brazil was because my brother and I started losing that language. We were surrounded by so much English that we started losing it a little bit, and then she decided, you know what, it's time to reconnect them a little bit with their culture. So we moved to Brazil for a couple of years. We lived in a couple of different cities there. And then we moved back to the US when I was 15. So I was able to do high school here. I moved around between a couple of different states at that time as well. And then for college, I decided to come back and go to Temple, where I'm still at in Philly. And that's really exciting. I think it's shaped me in a really particular way. Of course, growing up, I didn't really understand why all the moving and, going from one place to the other. And it felt like we sometimes didn't have a really stable, just life path. But now I'm really grateful because I feel like it's really opened up my eyes to see all these different cultures and how people live differently across the two different ends of the world, right? And, north and south. And so to me, that's really exciting. And nowadays in research, I think it's really exciting to be able to have that perspective and understand how even the same condition can affect people differently just based on where they are geographically.
Yeah, I'm kind of curious. Is there kind of a difference between the research community in Philly and the research community in Brazil, as best as you know? Like, what are some of the big differences that you think that you see?
Yeah, so I've never actually done research in Brazil, but now at this stage in my career, going to conferences, I've actually been able to connect and meet a lot of Brazilian researchers, and so that's really cool. We're able to finally like talk at this stage in our careers to talk about these big differences, and I think that the biggest difference I could probably think of is actually in funding. Some countries outside of the U.S., especially in countries in South America, they don't have a lot of funding, a lot of resources to do science. But then still they're out there and they're breaking records. So I think it's still pretty amazing, what passion alone, like how passion can take you so far. There's also a ton of groups actually that also work in Alzheimer's disease, particularly in the southern part of Brazil. And I love connecting with them every time I go to a conference and seeing what they're up to. And also, they tend to do a lot of research, also just looking at how that population in South America is impacted by different types of dementia. And so I think that is really impactful work.
Yeah. Yeah, you mentioned your career. Tell us where you're at in that career. What is it? What is your position? You're in Philly, you're at
Yeah.
Temple, but talk about where you are in that journey that you've been on and where you're going.
So I'm at a really exciting time right now. I feel like for the past 2.5 years, I was waiting to get to this point. I just recently became a PhD candidate. So I'm done with all of my classes, done with that first master's part of the PhD.
Congratulations.
Thank you so much. So at this stage, I'm really just focused on the science, which to me is the most exciting part, being able to go to lab and do experiments. So I'm a PhD student right now, PhD candidate, which is exciting because I get to have like all that taste for the lab and doing experiments and finding things out for the very first time, when you do an experiment and you find something that no one knows yet. So it's really exciting.
Yeah. Yeah. So, I think most people don't understand the time commitment as far as years now that you're a PhD candidate, how many years do you have in front of you before you start to be an independent, assistant professor? How many more years do you think that's going to take?
Yeah, that's a tough question. It really is a long commitment. We start in undergrad. So usually you have your first 4 years of undergrad, depending on your institution and your major, you're going to have some research requirements as well. And so I had to start research at that point. So I did a year of research in Dr. Fasadi's lab as well as an undergrad. And then from there, I spent a few months as a technician. Then I started my PhD. I'm currently in the half point of my third year, which means hopefully I have 2 more years ahead of me before I get my PhD. And then after that, it's postdoc. And then after that, maybe another postdoc, right? Depending on how my career path actually goes. And then finally becoming possibly an assistant professor and
Yeah,
starting my own lab, which is the goal.
that's amazing. It's a lot of years. It's a lot
Yeah.
of years. So, with that commitment and all that time and the excitement that you feel, why did you pick the brain? What is it that you were fascinated about? And particularly, what is it that you're researching when you go into your lab?
Right. So I picked the brain. I think I always had a really big interest in medicine. And then as I was growing up, of course, my grandmother had Alzheimer's disease, and I watched that trajectory with her. She was diagnosed the year that I was born, and she lived with the disease for 16 years. So I feel like I was able to see, like, well into my teenage years, how Alzheimer's was taking her away, right? And I would hear these stories. She never answered to me when I would call her grandma because she didn't know me, right? So she couldn't create those memories anymore. But people would tell me stories, and I would see how the disease was kind of taking her away. And that made me fascinated. I loved having conversations with her as much as I could. She had a very short attention span, of course. So our conversations lasted maybe a few minutes, and then she would completely lose track. She didn't know who I was. She didn't know what we were talking about precisely. But I liked having those few moments where I felt like I could get some insight into how her brain worked or how it wasn't working. And I think my fascination with neuroscience really started there. So I went to Temple. I chose a neuroscience major because I always had this intense focus in Alzheimer's disease. I think specifically, I really wanted to understand how it worked. And so during my undergrad years, I did have the amazing opportunity of of doing volunteer work at Abington Hospital, where I worked with geriatric patients, and I was able to see dementia up close with some other patients. But then I found a lab, and I think that's what made my heart beat a little bit faster. I've always wanted to see both sides of the story, the clinical side. I wanted to see how research worked. But inside of me, I thought for whatever reason that I would connect more with that clinical side, but it turns out that I just fell in love with basic science and just understanding at the molecular and cellular level how things work. So currently, that's what I do. I work with microRNAs, extracellular vesicles, just really tiny molecules overall.
Yeah, it's interesting because a lot of people know that, people go to school to become doctors or, people understand that there's researchers, but I don't think that a lot of people understand that there's people that go into research that were motivated because of medicine, thinking that they were going to become a doctor.
Yeah.
And do you think there ever be a time when that kind of pulls you back and you become an MD, PhD?
I don't think for me, there are a lot of people that do that, and I think that it's
Mhm.
amazing. Of course, we have a lot of researchers that work in that clinical side with, clinical trials and working actively with patients, and I think it's amazing work. I really do enjoy the basic biology side of things. So I think for me, it's probably just going to be the PhD path, but one of the things that I enjoy the most about when we go to scientific conferences is being able to speak to researchers across these different fields. And the reason why I like that is because I think it's so important to keep in mind, even when studying basic biology, that there's a reason that we're doing that. And the reason is because there are real patients, right? Real people that are suffering through these devastating diseases. And so I recognize that and I keep that at the back of my head at all times, that there's a purpose to what I'm doing and that there's an ultimate goal, right, to bring cures or early diagnoses and things like that. But as for myself, I think I want to stay closer to the side of just studying the basic biology side of things.
Yeah. So you mentioned these microRNAs. So
Yes.
what is an RNA? Tell me in your own words, because
Yeah.
other people have ways to describe it. And what is a microRNA?
Yeah. I think it's a great time to be studying them. So RNAs have gained a little bit of attention since 2020, the release of the messenger RNA vaccines. I think some people are a little bit scared of them, and I've heard comments from everywhere. People just don't really understand what an RNA is. I've even heard people saying that the COVID vaccine was injecting people with messenger RNAs, and I don't think people realize that messenger RNAs are such a huge part of who we are. They're everywhere. Messenger RNAs particularly produce proteins, right? So when we're talking about proteins and doing things and activity and cells and across cells, across organs, we're really talking about those messenger RNAs making proteins, starting these pathways. But then in 1993, we actually found out that there are a ton of other types of RNAs that don't just make proteins. So before that, we already knew we had tRNAs and rRNAs. And then we found also these non-coding RNAs that their purpose is actually not to make protein. They have the functions on their own. And microRNAs are just one of those types. So microRNAs, they're really tiny. They're about 20 nucleotides long. And what they do is they're able to influence cellular pathways through a complex called the RNA-induced silencing complex. So it's a protein complex where this microRNA will bind and be sort of a guide. Then it's able to physically attach to messenger RNAs. And then the RISC complex, those proteins surrounding it, lead to the degradation of that messenger RNA. So it's kind of a complicated process where you have like one thing canceling something else, and then you have an entire pathways being impacted by that. So then these microRNAs, that's what they do. They impact entire cellular pathways. They have multiple targets. And the reason why we're studying them is because, well, in terms of scientific field, it's such a new field because they were only discovered, a few years ago, if you really want to look at it that way. And there are millions of types of microRNAs everywhere across all organisms. It's a very conserved molecule. Um, and yeah, so they're now being studied across a multitude of different diseases. They have impacts everywhere. So yeah,
So you mentioned a couple words that you said they bind and they guide,
yes.
and then you said then they create cellular pathways. Are they binding and guiding to in a positive way for, to a positive cellular pathway where you have these other RNAs, creating the proteins and the things that we need for duplication of cells and all that kind of stuff?
Mhm.
Or is it a negative where it's creating kind of bad pathways into a disease? Or do we not know?
It's actually both. So we have, because we have such a variety of microRNAs, just like proteins, right? You have proteins that lead to certain pathways and other pathways. You have proteins that end up degrading other proteins, and then you impact the pathway that way. MicroRNAs, what they do is most of the time, there are some research now just starting to show that maybe they can have other functions as well, but the one that we're most familiar with is the fact that they're able to degrade messenger RNAs. So then they're preventing certain proteins from being made. And then those proteins that are not being made, those pathways are either going to cancel out or lead to a different pathway. So depending on the specific microRNA that you look at, you could be looking at something that's leading to a positive outcome or leading to a negative outcome. Or a balance, right? Because we always talk about the good and the bad, but I think if we bring in inflammation as an example, right? We talk about inflammation as is it a bad thing, right? But no, not really. Inflammation is a good thing depending on the context, but it could also be bad. So microRNAs are kind of in that same level. The microRNAs that I specifically study, which are microRNA 212 and 132, even within this very specific field, just studying these two microRNAs, there's a lot of diversity even because they're thought to be harmful for the heart. They cause hypertrophy. There are papers out there talking about that, but they're generally considered to be very protective for the brain. So then when we're looking at therapeutic approaches, we really have to take into account those two things, right? Because we want to be able to preserve the brain, we want it to be healthy, but we don't want to damage any other organ as well in the process. So it's really important to keep in mind that balance.
Yeah. Yeah, I was sitting there, I was thinking, and when you mentioned that, because you're in Dr. Fassetti,
Mhm.
Sylvia Fassetti's lab,
Yeah.
and that lab does a lot of blood-brain barrier, trying to understand
Mhm.
just the relationship between the cardiovascular and the nervous system, and particularly the brain. What is it specifically then that I know there's a lot going on in that lab, but what is it around these two microRNAs that you're trying to answer? Like, do you have a couple hypotheses that you're trying to test? What is it that you're specifically, you're trying to do?
Yeah, so I wanted to essentially my project started with trying to understand the physiological roles of these microRNAs. The reason for that is because microRNA 212 and 132 have consistently been shown to be downregulated in the brains postmortem of Alzheimer's disease patients, as well as in the plasma. And they
And what
seem
is
to correlate
what does regulated
downregulated.
mean? Yeah.
Yeah, it just means that they're being degraded. So you have less of them compared to healthy age-matched controls. So then we wanted to understand why is that, right? And also, there's literature out there showing that as Alzheimer's disease progresses, so as you have like a progression of Alzheimer's, you have a progressive downregulation of these microRNAs. So then we wanted to understand, is there some sort of pathway that they're involved in that is causing the disease, or is the disease causing this downregulation, and what's their function? Because we didn't really know that. So a few groups have been studying microRNA, these specific microRNAs also in neurons for a couple of years, and we found some specific proteins that they target that actually do lead to more Alzheimer's disease. So essentially, when you have less of these microRNAs, you have more pathological outcomes of Alzheimer's disease. But what we found is that we didn't really know what was happening in endothelial cells specifically. So endothelial cells, they line the blood-brain barrier. So they're the first layer of protection between the blood and the brain. So our group looks at endothelial cells very deeply because we want to understand these mechanisms, right? What can be disturbed there? What is causing the rupture in the blood-brain barrier, which is an early event also in Alzheimer's disease. And in my situation, I really wanted to understand what these microRNAs were doing in these cells, how they were impacted by amyloid beta, and how they were impacting the progression of the disease in those cells as well. So I wanted to look at that. That was, I guess, the first step. We wanted to look at how they were regulated and what they did. But then as I read more and got deeper into the literature, we also realized that these microRNAs are released in extracellular vesicles, meaning that the cells release them encapsulated in these extracellular vesicles that are released into the blood, which allows them then to be preserved in the blood for quite some time. So then we wanted to be able to see, so if we got the blood, could we detect these microRNAs as well? And if so, then would we be able to detect this dysregulation earlier on in the disease, because it is something that happens earlier on. And so right now, one of the biggest goals of my project is to identify not just microRNA 212 and 132, but also a panel of microRNAs that show this dysregulation earlier on. And like I mentioned, I study mostly everything in the context of amyloid beta. Which is why our lab also focuses a lot on cerebral amyloid angiopathy, which is the accumulation of amyloid beta specifically around the microvessels in the brain.
So, if I understood you correctly, then when you said detect this earlier, that could possibly lead to some sort of blood test younger in humans to be able to see if this downregulation is happening, that would start to lead towards understanding that this person may be in a higher probability for Alzheimer's. Did I understand that correctly?
Yes, yes. So I think right now, as we stand in research, we already know that Alzheimer's disease starts developing about 20 years before we have manifestations of the disease. So, meaning 20 years before you're forgetting where you put your keys or you're forgetting who this person is, you already have the pathology starting. But one of the biggest issues that we have in the clinic is that we're not able to actually diagnose what person will develop Alzheimer's in 20 years. So we have risk factors. We know certain life situations that you could be in that could lead you there. Of course, we know a lot of the genetic factors that could lead you there. But most factors that end up leading to Alzheimer's disease are not genetic. However, we still have these indications of disease. We're just not able to track them yet. Even to be able to detect and diagnose Alzheimer's disease today, most of the time you would have to probably do an exam in your brain to be able to detect amyloid. But not every Alzheimer's disease patient has amyloid as well. So Alzheimer's is a very diverse disease, and I think that's why it's important to be able to separate also these, who has just Alzheimer's or who has also cerebral amyloid angiopathy, who has this and that, so that we're able to diagnose and then treat according to what that patient specifically needs. So really going into personalized medicine. So that's one of our biggest challenges. And that's why I'm very interested in being able to develop a panel so that we could be able to track specifically cerebral amyloid angiopathy earlier on, and then with that be able to prevent the disease from progressing in the first place.
Yeah. Yeah. So, you received a grant from the Karen Toffler Charitable Trust. For anybody that's out there that is wanting to give to researchers, there's really kind of an importance to that, especially in where you're at in your career. Can
Yeah.
you, what does this mean to you, and how does that support not only the science, but you as a person trying to become an independent researcher and stay in the field?
It has a huge impact. First of all, because everything in science is very expensive. So we really can use all the help that we can get. Um, but I think as an early career scientist specifically, I think it's the kind of award that really gives you the confidence to move forward. So not even just the financial amount, but also even opportunities like this to raise awareness to these issues that we're studying, so that the population can also know what we're looking at in the labs and how hard we're working to be able to find answers to all these unanswered questions. So I think having foundations, or just really anybody who has the time to listen to what we're doing, people who can invest in research, I think it's so important. It gives us the confidence to continue. It gives us the financial assistance to continue, which is also extremely important. So yeah, I'm very, very grateful for the award and to be a Toffler Scholar.
Yeah, it's amazing because there's not a lot of funders that fund this kind of cohort as far as your um, it's not age group because some people go into research later. It's really kind of that place in your journey where you're
Yeah.
in an important pivot point. And you mentioned it doesn't take a lot of money. It doesn't take a lot of money to be able to pay for some time on a microscope or go to a conference or share. Is there anything in particular that this money for you is going to allow you to do that maybe you wouldn't have been able to do?
Yeah, I mean, well, conferences for sure. A part of the aid package that I received also allows me to go to one conference during this year, and I'm so grateful for that because it's really important, I think, in my opinion, to go to conferences, to talk to other scientists, to kind of integrate what everybody is doing. Because I'm studying such a small and specific part of the disease, and but it's like I mentioned, it's really important to keep in mind that other people are doing other things and to be able to integrate that, right? So I think that's one thing that I'm really grateful for, being able to go to conferences this year. Um, Also, very recently, I actually just started this new set of experiment with RNA sequencing, which is a really cool technique that allows us to see the entire transcriptome within a cell under a given condition. So this money is also sponsoring that, and I'm really, really grateful for it. It was something that most likely I would not have been able to do, so I am really grateful for that.
Yeah, that's great. It's so interesting that you said that that these microRNAs have just it's only been 5, 10 years that we even knew that they existed, and it's only been a couple decades really since we mapped the genome,
Yeah.
and there's really an acceleration. What it's really great to have younger voices, kind of talk about what research is like and what do you see for the future? Do you're part of that acceleration. How is that um, you know, affecting how you think about it, especially in institutions that may be a little slower?
Mhm.
Like what is that dynamic that you see?
So we've known for microRNA specifically, we've known about them for about not even 30 years. No, sorry, 2026. 30 years, 33 years. But it's still a newer field because if we really consider where science started, right, so many years ago, and there's so many different aspects like proteins that we've known for such a long time. And still their entire studies focused on studying that, right? So I think even though it's been a couple of years now, it's still a really fresh topic to be studying, especially in such detail. But you're absolutely right. Science has been advancing to a point where now we have extremely cool techniques that allow us to visualize things that perhaps scientists couldn't even imagine many years ago. And I think for me, starting my career already in this environment where we can do all these cool things, it makes me really excited. I think it's a really cool time because you can have a question and you have a larger possibility today of actually answering that question, and not having to map the genome by yourself, and not having to do all of these things that amazing people have had to do in the past. So it's really exciting, but it's also really challenging because you're stepping into a growing field. You're stepping into something that has already been so advanced. And nowadays, with all of the omics studies, so omics is really when you're studying the entirety of anything. So you could have proteomics where you're looking at all of the proteins within a given organism or within a given cell, or transcriptomics, which is what I do a little bit more, where you're looking at all of the transcripts, all the RNAs within a cell. Um, it's really, I think it's challenging to know the direction to go from there because you're looking at such a huge data set, right? And you're trying to pinpoint what's important, what's not important. You don't want to overlook anything. You want to be able to go in the right direction. So that's a little bit challenging, but I also think that with all the literature we have out there, nowadays also we make a lot of these databases public so that other people can access and not have to repeat the experiment, but rather just reanalyze something that someone else has already done. I think it really brings that integration also in a way, where we're all looking at the same data sets, not the same, but similar enough data sets, and trying to get to the same endpoint. So yeah, I think it's a really exciting time to be in science, but I also think it is pretty challenging to step into something where so many people have done so many amazing things already.
Yeah. How is technology helping you with all that data? The omics, it's just a lot of data. It's a lot of maybe understanding how all these pieces and part relate to each other. Is there, do you have to be fluent in using AI and, some machine learning? What does the lab, look like, right now as far as helping you with that?
Yeah, so I think in my lab, because we are in a wet lab, we don't have all of the precise techniques to be able to look at that. So we do a lot of collaborations with people that are more in bioinformatics and those kinds of labs. But I think nowadays perhaps it's a little bit more important to learn even how to do a little bit of programming, or in regardless what language, a lot of scientists, a lot of people that graduated with me even have learned Python. I personally had to learn a little bit of R as a requirement for my school, which is also a programming language. And then now with the emergence of AI, I think a lot of people are skeptical. And usually in these conferences that we go to, we always have one or two talk sessions about artificial intelligence and how to use it for to help us really analyze these data sets, but without giving too much away or without um, how do I say it, without making AI also substitute our jobs, right? And doing it in an ethical way where we're still thinking, we're still coming up with these answers, but we're using AI as a tool to maybe organize data sets or to maybe help us pinpoint what's important. I personally don't use any big AI data sets or machine learning to help me analyze, but like I mentioned, our lab does a lot of collaborations with people in bioinformatics and stuff.
Yeah. Yeah, it seems to me that you know, I think there's a lot of hype with AI that but if you allow the AI and you really don't go into the lab and do the experiments yourself, how do you check
Yeah.
the AI's data? That's what I heard you say.
Yes, exactly. There's a lot of checking for sure. I think it's a really, it's a very, I think, controversial perhaps topic.
Yeah.
Now we have AI doing things that we couldn't even imagine, or we still don't understand, right, how machine learning even actually works if we're really looking at it. And that's probably because we also don't understand how our brains work entirely, right? So we've created something to kind of model how we work. Without understanding either side, so to fully, so yeah, it's a very controversial topic, so I think just being, really careful with it, but also using it as a tool, and it's doesn't seem to be going anywhere. So
Yeah. Yeah, that's really funny that you said that we model our we model AI after how we think or we problem solve, but we really don't know inside our brains how that really works.
exactly, yeah,
We really don't understand. We don't understand where it comes from. Yeah, that's interesting. So
yeah,
back to being in your lab. I know Dr. Fassetti has won these awards on mentorship, and she seems like she's very collaborative.
yes.
What are the leadership experiences that you're getting the chance for you to mentor and you to do? Tell me about that.
So Dr. Fasadi, she's great, honestly. To me, she's been my PI for a couple of years now, so we've had quite a long journey together. I started in her lab as an undergraduate student. She then hired me as a tech, so I was constantly jumping from projects. So for my undergrad project and for a tech, and then I started my PhD program, and I actually only applied to Temple. It was the only school I applied for because I wanted to stay in her lab if possible. So she really is a great mentor. And I think that, at least for me, my trajectory really, really reflects that. And I feel like she's always had a very specific way of helping me grow. And I can see that, and I can see how much I've grown over these past couple of years. Now, specifically as a PhD student, she gives me not only the opportunities to go to these conferences, sometimes even alone, but she also, she helps us with networking, with meeting a lot of people. She's very well connected. And then recently, also over these past 2 years or so, I've been getting a couple of students that I can then mentor. And it really brings me back to like a full circle moment because I was also an undergraduate student being mentored by someone. And right now, I currently have my second student, and she's an undergraduate. I had a master's student before her, and I think it's really exciting. We're starting up a new project for her, and it kind of reminds me of when I started my first project in the lab. So it is exciting.
Yeah. And they don't give you guys a lot of leadership training, do they? You have to kind of pick that up from your PIs and people around you. Do I have that correct?
I think it depends a lot on your program. So I'm at Lewis Katz. Temple has two separate programs. So I'm at Lewis Katz School of Medicine in the biomedical sciences program, and we actually don't have a teaching requirement, which is it is good. I consider it a good thing. It allows us to focus full time on research. But we do have a lot of collaboration with the undergraduate school as well, with Temple. And they send a lot of students, which then we have the opportunity of meeting and mentoring some of them. So I think the whole collaboration, Temple is an extremely collaborative school in general. So I think that just really bodes well for the program as a whole because it gives us these opportunities without it being a requirement. So then it allows us to really grow in the areas that we want to grow in. Of course, not everyone likes mentoring. I personally love it, but yeah.
Well, and it takes a lot of communication, which, communicating science and communicating especially the science that you work on is really hard. And it's very important. How do you learn to kind of break this down in a simple, because you talked about the microRNAs in a very simple term. I understood what you were saying, but is that sometimes a challenge to take science and then communicate it in a simple way that normal
Yeah.
people, non-scientists can understand?
Yeah, I mean, sometimes even scientists as well. I had a meeting earlier today where I was talking about a specific topic and I was talking about exosomes for an upcoming presentation that I have. And the person who's working with me on this project said, just remember to include a slide or two about what exosomes are because, and this is for a group of scientists, but because sometimes we do take for granted what people know and what they don't know. Not that there's any shame at all in not knowing. I think personally, I have a passion for explaining science, and I try always to explain it in a way that everyone can understand because I think scientific advances are for everyone. It's not just for scientists. So, like even pain medication that we take, right? Like someone had to research that at some point and make it available to the community. And so I firmly believe that all of science should be available to the community, which is why I try very hard to always explain things in very simple terms and see that people are going to understand what I'm saying because it's also important for them, hopefully.
Yeah.
Um, but yeah, sometimes there's that challenge, especially in mentoring, because when a student comes in, you don't really know, what they don't know. So for me, I try to be very open with my students about that. I say if I'm saying something you don't understand, tell me. Don't be ashamed. There's absolutely no shame in not knowing. And I'll try to explain it in a different way because I feel like once you've been working on something for a few years, in your head, everyone knows about that, and it's no, people don't know. So sometimes even within the scientific community, people are not going to know what you're talking about. So it's really important to be able to break down because we want everyone to understand, right? We want to be on the same page. So, yeah.
Yeah. That makes me feel better that everybody that even scientists have some confusion with some of the terminology
Hmm.
but I love that science is for everybody and I do believe especially in today's environment where there's a lot of responsibility on people to understand and or to be able to communicate with their doctors and ask the appropriate questions because many years ago, you just took whatever the doctor said.
Yeah.
And now there's so much information out there. How do you see that changing, especially around, cognitive decline, Alzheimer's, maybe you reflect on what your family had to go through with your grandmother, just being able to ask what is going on.
Yeah, I think it's important, and also I believe it would help if people understood, um, just not even just Alzheimer's, but a multitude of diseases that we have nowadays. I think if we were all able to do the right research and understand what is going on, what we do know and what we don't know about specific diseases, I think it would help us break that barrier and trust that people have in science. So not to bring it back, but already bringing back the COVID vaccines, this is a conversation that we had a lot with, just amongst ourselves, that in part to see how the population is so afraid of vaccinations now is in part our fault because we didn't explain them well enough. So to me, it's really important to have that open communication about science. If there's something we do understand or something we don't understand, and of course, there are all those conspiracy theories about scientists knowing the cure to all diseases, and it's sometimes it's just not true. Like we wish we did. We don't know. But I think if we had more open communication about what we do know, what we do understand, what is a challenge? Why don't we understand how this happens? Or what do we know that could maybe help us, take a step forward? I think that's really important for both sides. It's important for people in science because maybe that would help us have, just a little bit more of the trust from the population, a little bit more funding, perhaps a little bit more everything. But it would also help the everyday person who's just going to the doctor and trying to understand what their exams mean, and how their health is going and how to move forward. It also, I think, would be such a huge leap towards personalized medicine, which is something that my heart is really into because I think that people are complex, people are very different. So even if you take one disease like Alzheimer's disease and you see that not every person with Alzheimer's has the same pathology. So I can't just create this drug and give it across the board because it wouldn't work for everyone. And in some cases, it might be even more harmful for someone. But that would only work if we had patients that also understood a little bit about their own health. So just, personally, I'm very, very passionate about spreading science for that reason, so that people can have this basic understanding, because it is their bodies, right? So to have this basic understanding of how we work as human beings.
Yeah, it's amazing. We don't know. Most people don't know what goes on in their body. They just don't know. They don't
Yeah.
know. And then they hear things. I get it all the time. Like, well, why haven't we cured cancer? Like, well, there's dozens of different cancers, and they
Yeah.
all are different. They're,
And there
you
are
know,
cures as well. That's something I always like to bring up because when people hear that you're a scientist, that's something that they say. They're like, "Oh, is it true that you guys know the cure to cancer and it hasn't been released?" And because it makes money. And to me, it's so, I always joke around and I'm like, who's making money out of people dying? It doesn't,
right?
no, we don't make any money out of it. If anything, if we kept people alive longer, you would probably get sick of something else and then we would make more money, right? But so I try to explain, like, well, maybe if we take a step back and understand what cancer is, right? Maybe we would understand why we don't have a cure for it. Or even looking at the fact that we do have a lot of therapeutic approaches nowadays that do cure cancer. So to get people kind of on that same page about we do have these advances, right? People ask me sometimes about why we still see this rise in Alzheimer's disease cases every year. And I say, well, it's a medical miracle because we see the rise because we see the population is getting older, which is a good thing, right? We're living longer, and therefore we have the opportunity of developing more diseases that we didn't know of beforehand. So now we're getting to that older age. Now we're seeing that we have these other diseases, and now we're trying to find the answers for those as well.
Yeah. Well, and we also have a name for it. Many years ago, we didn't have a name for it, and so it was probably just dismissed.
Yeah.
Now we have a name for it. We know, we know some of the what of it. A
Hmm.
lot of people don't understand the why and why is this happening, and especially in individuals. You've mentioned a couple of times about you're just really passionate about the personalized medicine side of this. Say more about that, and what do you hope to achieve in 10, 15 years as far as that is concerned?
Yeah, so when I say personalized medicine, I try to encompass not only just the genetic side of things, because of course genetically we're all 100% unique, right? So that's a big factor already. That already means that something that could work for me may not work for someone else. But aside from that, we also have different pathologies. We have even, where you live impacts you so much in terms of your health and your brain health specifically. So when we're looking across the board, particularly into what I study, particularly Alzheimer's disease, we're looking at not just one condition, we're looking at a multitude of diseases combined into one. Um, so the reason why when I say that I'm trying to develop this biomarker panel, it's mostly for cerebral amyloid angiopathy because we already have a lot of biomarkers for Alzheimer's disease. We might, it would be, of course, beneficial to be able to find more, and especially that could diagnose the disease earlier on. But I think it's so important to also be able to differentiate who has what is purely Alzheimer's, because as of right now, the clinical definition of Alzheimer's disease is people that have amyloid beta plaques and tau tangles. But not everyone has amyloid beta. Not everyone has tau. Some people have tau and amyloid and don't have a cognitive decline. So then is that even Alzheimer's, right? So we have all these different aspects of the disease. We have vascular dementia. We have vascular contributions to cognitive impairment. And we have CAA, which is cerebral amyloid angiopathy. So when I'm trying to develop something so specific to such a specific part of the disease, it's to help integrate that into personalized medicine to say, okay, this patient has amyloid and it has tau, but they also have vascular impairment. They also have amyloid around the vasculature. They also have this or that, which would help us then be able to come up with a clear treatment option for that person. Because as I mentioned, not every drug or treatment is going to apply to every single person. So it's really important to be able to take the actual patient into account and see what could be beneficial for that person in terms of therapeutics specifically.
Yeah. So many problems, so many questions, so many different aspects here. So when you look out 10, 15 years, what do you think? Where do you think the research will be? What do you think we may have achieved? You're not going to be held responsible for any of it because it's a what if question. But
Yeah.
what do you think? What do you think we might? Where do you think we might be with all of this?
It's a really hard question to answer because, like you've mentioned, right, science is moving so fast. And it's getting progressively, exponentially faster, right? So when we look at what happened in these past 15 years, we can see so much advancement already. So many things that we believed in 15 years ago are no longer true because now we know better, or now we know the pathway deeper, or we know something extra that impacts how we overview the whole thing. Even within the past couple of years within Alzheimer's disease, we started seeing how important the vasculature is and how important inflammation is in the brain during the progression of the pathology. A couple of years ago, we thought amyloid was driving the whole thing, and now we see that, well, maybe it's not. Maybe we have these other things also driving these conditions. So I think it's really hard to answer where we could be in 15 years. Of course, within the past 15 years, we've come up with drugs already, the first drugs, anti-amyloid drugs for Alzheimer's disease that aimed to actually stop the progression. So I think, if we look at the advancements we've already made, I think we can be really hopeful to where we will be in 15, 20 years, especially if we have appropriate funding, if we're asking the right questions, we have enough people looking into these questions, which Alzheimer's is a pretty good big field. So I think I'm really hopeful to where we will be. Hopefully we'll be at a place where we start having a decline in the amount of people getting Alzheimer's disease, regardless of an aging population.
No. Well, what gives me hope is, seeing young people like you and young career researchers so passionate about asking these questions and, dedicating your time and your energy for all of us. And so, seeing your passion show through and listening to it is that gives me hope for the future because you're right. These things are devastating for not only the individual, but for the family, and not to mention the economics of it. So I appreciate you and what you're doing very, very much. much so. Is there
Thank
a,
you.
yeah, is there a question that you wished I would have asked you that you wanted to just kind of get some message out or let people know about that I didn't ask?
Uh, Not that I can think of right now. I
Okay.
think, yeah, I really liked the direction in which the questions went. I think it was really fun.
Oh, good, good. That's the main purpose of these. So, if somebody's listening to this and they want to get a hold of you and find out what you're doing, or maybe even see if you need some support, how do they get a hold of you?
Yeah, so I think for my career, the best way to probably follow along to what I'm doing is through LinkedIn, just Micaelly Alves on LinkedIn. If anyone has any specific question they would like to contact me, my email is micaelly.alves@temple.edu. And I'll be happy to answer any questions. I love talking about science. I love talking about the brain. It's absolutely my favorite subject. So yeah,
Yeah, I sure appreciate you. Thank you so much.
thank you.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.